evening, everyone. How's everybody doing? You're wasting valuable studying time by coming and listening to this.
Wunderbar.
What's that?
Wunderbar.
So hopefully this is providing you more than you would get if you were studying. So if it's not, then obviously you're wasting your time here, aren't you? I never thought about that before, but it seems like this has to, the pressure is on me now to be productive. So if it's not, then you have someone to blame for why this isn't working. So how's studying going? So the good news is that with that flood day, we actually have about one less day of material on this exam than we usually have. So in that sense, it is a lighter exam. My exams, as you know, always have a lot of material on them. So that's, I guess, a bit of good news, but it means that it'll catch up with us later, and it probably will catch up with us on the second exam. So second exam will probably have a lot of material on it, is my gut feeling. This will be like I've done before, so I'll be here answering questions as long as you would like, and we'll go from there. I will say if you haven't looked at the practice exam, you should. Okay, practice exam is, the format of the exam is different this term than it was last term. In fact, it's significantly different. So I would encourage you to look at the practice exam and make sure you read the instructions carefully. One of the things that students mess up with on this exam is not reading instructions carefully, particularly for the first section. So make sure you understand what's in that first section. Okay. I meant to mention in class today, and I didn't, and I'll try to put this out in an email to everybody tonight, but the exam has, as I recall, 20 points in the first section, 50 points in the second section, and 30 points in the third section. And there is an extra credit question on there for you for singing loudly. I liked the singing today. That was kind of good. All right, so where are we at? What's up? Giovanna? These are all academic questions. We're all academics.
Looking at the Pyruvate dehydrogenase.
Yes?
In one of the figures, you described it as being E1, E2, E3.
Yeah.
I have it here. I can show you. But actually, when you're going through the processes, it's not that, because the E3 that you're describing is actually just reforms.
Yeah, a lot of people have concerns about what I said about E1, E2, E3.
I understand.
No, that's fine. No, I have no problem with that. I'm happy to. So I'm glad you asked the question, because that gives me a chance to just point out some stuff. One of the things that happens here is you have 3 subunits, and you have things happening between them. So we don't have this thing where we have 3 enzymes. Enzyme A passes off to a separate enzyme B, passes off to a separate enzyme C, which is what we're used to thinking about with enzymes. This is all happening in the confines of an enzyme. Okay? So where we say exactly this happens, and where we say exactly this happens, and where we say exactly this, words are going to fail us. And that's part of what you're seeing, the difference between what I'm telling you and what the book tells you. I will say, and I've pointed out to some of you who've seen this, or who've sent me this, that this schematic that the book gives actually is completely consistent with what I said in words. And so my word descriptions actually come from this figure. Okay? And I think this figure is a very accurate description of what's happening in that overall process. So as I said in class, I'm not really obsessed with E1, E2, E3. I don't honestly think that they have that much relevance to us. I think that the important thing is what's happening in this process. And if we call what happens, a decarboxylation in E1, okay, but that isn't the important thing. The important thing is that a decarboxylation happens. And then an activated intermediate is formed. And that activated intermediate is on TPP. Whether we say that's E1, E2, E3, that's really not important for our purposes. Okay? And I really want to emphasize that. But I do think that the steps in that mechanism that I've talked about are important. Okay? So you have a decarboxylation. That decarboxylation, if we're talking about yeast or bacteria, can be a stopping point, or actually a branch point, to making ethanol if they don't have oxygen. That is, they have no NAD. In our cells, we don't have that option. And in bacteria and yeast who have oxygen, they don't stop there. They continue along the process. And that decarboxylation creates a reactive intermediate called activated acetaldehyde, and that simply gets passed to a lipoamide residue. Okay? So that lipoamide residue is a carrier of that reactive acetaldehyde, and it simply takes that reactive acetaldehyde and passes it off to lipoamide. So it's just a pass that's happening. That pass happens to lipoamide, and it's at the transfer to lipoamide where that oxidation occurs. And somebody came and asked me the other day, is the oxidation exactly where is that oxidation? And is it, is it as it's being passed, or is it after it's on here, or whatever? The oxidation is happening as it's transferred. As it's transferred, we see that this is going now to a thioester, where before it was an aldehyde. Actually, it was a reduced aldehyde up here, but this is definitely an oxidation that's happened. That's happened on the attachment of this guy to the lipoamide. That meant, and you can also see that by the fact that the single bond, which was the the disulfide bond that was there, has been reduced, which is also evidence that oxidation of this guy has happened because it's given up electrons to these two sulfurs. The two sulfurs now are reduced. This guy gets passed off to CoA. That's not an oxidation. This is simply a transfer of this guy from one sulfur thioester to another sulfur thioester bond. And then everything else is just recycling back. This guy is reduced. It passes its electrons off to FAD. FAD passes its electrons off to NAD, and we're back where we started. So those steps are what matter. Where we say E1, E2, and E3 fit in really doesn't matter. Does that hopefully clarify for you? And I sort of apologize for bringing up E1, E2, E3. In previous years, I haven't done that. And it's one of those things you learn in teaching is that when you give people more information than what they need, they do get worried about that information. And that was my fault. My phone is telling me something. Oh. Sorry. I like this message. It says, "Hi, I'm in town and I have a pie for you. Can I bring it by?" Damn. Why did I schedule this? Yes, back here.
Are you expecting us to know the structures of the molecules in the citric acid cycle? Or?
Okay, so I have addressed that in the lecture. So if you look back on the... Okay. Well, again, don't study practice exams. Learn what I've talked about. So, okay? Yeah.
Can you go over how is the net synthesis of glucosinate with the glyoxylate?
Yeah, certainly.
Where does the extra carbon come from?
Yeah, sure. It's a good question. Let me get you that. So let's go to the glyoxylate cycle. And where is the glyoxylate cycle? Right there, the very bottom thing, yeah. Thank you. You can stare at it and your mind doesn't tell you. So the glyoxylate cycle, as I said, overlaps with the citric acid cycle in plants, yeast, and bacteria. Okay? And by overlap, all I mean is that some of the same enzymes are common between the two. If we look at citrate synthase, it's common for the two. We look at aconitase, it's common for the two. And if we look at the enzymes that convert succinate over to malate, they're not shown on here, but those same enzymes that are in the citric acid cycle convert succinate to succinyl-CoA. Okay, I'm sorry, succinate to fumarate, fumarate to malate, malate to oxaloacetate. Those enzymes are common as well. The difference for the glyoxylate cycle is it has these two enzymes here that short-circuit the citric acid cycle. So by short-circuiting it, isocitrate lyase and malate synthase are avoiding the decarboxylations that happen in the citric acid cycle. So in the citric acid cycle, when we get to isocitrate, the next step is to make alpha-ketoglutarate with isocitrate dehydrogenase. Okay? Well, this is bypassing that. It's taking this isocitrate and making something else out of it. What's it making? It's making a glyoxylate, which is what gives the cycle its name, and it's making a succinate. Now, what this means is that we started with 6 carbons here. We had 6 carbons here, 6 carbons here. Now we've got 4 carbons here plus 2 carbons here. We haven't lost anything. In the citric acid cycle, we would have lost 1 carbon, and then we would have lost another carbon to get to this guy. So those 2 carbons are essentially saved right here. Okay? Well, these two carbons actually become useful to us because now the cycle can take this second enzyme and add a second acetyl-CoA, and in doing so, make malate and then oxaloacetate. Now, your question had to do with how do we have net synthesis of glucose, and the answer is as follows. If we go around the circle, all right, we start with one oxaloacetate and we end up with one oxaloacetate. That's just like the citric acid cycle. However, we haven't accounted for this guy. This guy can be made into fumarate, malate, oxaloacetate, and now we've got an extra one. So it's that extra oxaloacetate that's used to make glucose. We don't have the luxury of that extra oxaloacetate in the citric acid cycle because we start with one, we add two carbons, we get two decarboxylations, and we end up with one oxaloacetate. Does that answer your question? Good. Yes, Liz.
I feel like this is kind of a stupid question, but why is that?
You know, about a third of the questions I get are start with that very thing right there. So.
Why is that advantageous? Like, why do you want to make it into an extra? Doesn't that just go back through the citric acid cycle and stuff again?
Well, it's a good question. It depends on cellular needs. So I was talking to somebody in my office the other day about this. And this gives the bacteria, plant, and yeast cells flexibility that we don't have. All right? Now, for yeast and bacteria, that's probably not a big deal. But for plants, it could be a real big deal. All right? Why is that? Well, plants need a lot of glucose, not just for storage, but they use it to make cellulose. So now you've got an extra means of making glucose that you didn't have before. So you could, for example, live off of the oil that you've made and make glucose out of it very readily. We can't make glucose out of fat. Fat is our storage medium, and the only thing we can do with glucose is burn it in the citric acid cycle or make ketone bodies. We can't make glucose out of it. Okay? So this gives these guys much more flexibility in that process. So, yes, sir.
So my question has to do with the sodium-potassium channel selectivity.
Okay.
It's really just out of curiosity. So since in neurotransmission, the sodium channels open first.
Yes.
They are the smaller of the two channels.
Mm-hmm.
Would I be correct in saying that?
That's correct.
So that's convenient because they don't even have to deal with...
Don't even have to deal with it. Excellent. Yep.
So theoretically, or I guess hypothetically, if the two channels were switched in order,
Yep.
would the potential energy of the electrochemical gradient be sufficient to overcome the unfavorable energy of desolvation and resolvation of the sodium going through the potassium channel?
You know, that's a very good question. I probably won't have a very satisfying answer for you, but I'll wing it. Which is, I'm a professor. I get paid to wing it, so I'll wing it for you. I will wager that if you had it reversed, let's say that it were the potassium channels that were opening first, you would have not quite as clean of a signal. And part of that is, and I didn't really emphasize this in class, but more sodium ions leak in through the potassium channel than potassium ions leak in through the sodium channel. That sodium channel, because it is size exclusion almost purely, makes, I don't know, it's like 1,000,000 to 1, okay, selectivity for sodium. If you look at the selectivity of the potassium channel against sodium, it's about 100 to 1 to 1,000 to 1. So as you can see, you get a lot more sodium in through that potassium channel. And what that would do would mean that would really confuse and counter that gradient going the other direction. So I think it would have at the very least a small effect, but it could add up and make a larger effect. So that's what I would say based on what I know about the channels. Yes, Connie.
Going along the same topic, I guess I'm kind of confused how opening up sodium and potassium gradients will allow the information
information will be transmitted along the cell?
Okay, so our question is, how does opening of these channels allow information to be transmitted along the cell? Okay, let's imagine, I guess I'll just draw a little thing here on the board. Let's imagine I have this nerve cell here, and here's the end where all the action starts. Okay, so I see when the very first thing that happens is I open the sodium channels, and the sodium comes rushing in. Right? And that changes the electrochemical potential and causes now the potassium, which is in here, those channels to open up and the potassium to rush out. Right? What that creates at this end of this guy right here, what it creates is a high concentration of sodium and a low concentration or a lower concentration of potassium, where up here they're already low sodium and high potassium. Well, what's going to happen when we have a difference between the two? Diffusion is going to happen. All right? So we're going to see sodium, we're going to see potassium moving this way. We're going to see sodium moving this way. And that's going to change the electrochemical environment over here, for example, as it moves down. So we see a wave of voltage change moving down that nerve.
Okay, so the fact that like potassium is diffusing that within the cell is causing like channels to open?
So, well, just hold on. What's happening as the diffusion is happening is you're seeing a voltage change. Do you agree with that?
Yes.
Okay. So we see a voltage change moving across here.
Yes.
All right? The voltage change is what opens the gates downstream.
Okay.
So the thing that opened the gates down here, let's say it was we had a touch thing. Okay? So that fired the end of that nerve cell, but everything else along here, all these gates along the side of the thing are sensitive to voltage changes. They're exquisitely sensitive to voltage changes. So the slightest voltage change occurring as a result of this wave causes a new wave to start, and it goes all the way back.
Okay.
That's how it happens. Does that make sense? Liz?
Does the sodium potassium ATPase keep running?
Yeah, good question. Does the sodium potassium ATPase keep running during that? And it does. Okay, the gates opening up allow things to move faster than that potassium sodium ATPase keeps up. And these will close fairly quickly. Okay, this doesn't stay open for very long because it would be counterproductive for the cell to do that because you're going to, putting it out. It's kind of like the feudal cycle, right? We've got the not feudal cycle, the uncoupled mitochondria, where we have the protons coming in and we have the electron transport pumping them out, right? So exactly the same thing would happen if this were open for any period of time. Yes, sir.
Do both the sodium and potassium channels have that three-phase gated ball and channel motif?
They don't use the ball and channel thing, no. There are some things that do that. But they do have a pretty specific gate that's there, yes. Yes. Yes?
Not with nerve transmission, but with the E-type ATPase with the sodium potassium ATPase.
Yep.
It says that the movement of sodium and potassium is essential for the cell being able to maintain osmotic balance. Can you go over how that
Yep.
works exactly?
Yeah, so it's a good question also. So when we look at a cell, okay, I talked a little bit in class about how you do the experiment in a basic biology class where you take a dialysis tube and you fill it with, let's say, protein of some sort. And that protein is big enough that it can't pass out through the dialysis tubing. You close off the ends, you put it in water, and what you'll see is that the tube will swell, 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 because what's happening is water is diffusing in to dilute out the protein, but the protein can't come out. So if you let that go long enough, what will happen is you'll actually burst the membrane, because until you burst the membrane, there's no way of equalizing that pressure. That pressure in that example is very analogous to what's happening inside of cells. If we think about what's happening inside of cells, cells are, in fact, membranous or like that dialysis tubing. Water can move across their lipid bilayer very readily. It doesn't have problems like ions, like sodium or potassium or protons. Those have trouble moving across the lipid bilayer. But in fact, water moves across as readily as anything. There's 4 things that move across the bilayer easily: water, carbon dioxide, carbon monoxide, and what's the 4th one?
Is it NO?
I'm sorry?
Nitrous
Nitrous oxide will as well, but that's not the one I'm thinking of. Water, carbon monoxide, carbon dioxide, and oxygen. Oxygen, yeah, oxygen. If oxygen didn't, we're in trouble. Okay, so all 4 of those move across the membrane very readily. Well, water's the problematic one because water is the liquid one of that mess. So if we don't have some sort of trick to balance the pressure so that water doesn't want to diffuse in, water will and the cell will burst. And so the sodium-potassium imbalance that's created by those gates is one of the ways of dealing with that osmotic balance. And the mechanism of that is way beyond what we would talk about in this class. But it's just an example of a way to balance an osmotic pressure. Does that help?
It's a way that it tricks it.
It is a way, if you want to say tricks it, yeah, that's fine, yeah. Yes, Jody?
So with tetrodotoxin affecting sodium gates, if
Yes.
there was enough of that in the system, will that eventually lead to osmolysis of the cell?
So if you had osmolysis of the cell, no, I don't think so, because tetrodotoxin is a neurotoxin, and the sodium gates are for letting sodium back in. That's not the pump. So don't confuse the pump with the gates. They're different things. Yeah. So tetrodotoxin kills you because it kills your nerves. Yes?
Can you talk a little bit about liposomes and kind of how they work?
Yeah, sure. Liposomes. Let me get you a figure. I like that figure that your book shows of liposomes. So that was lipids and membranes. And where are we at? Anybody see it? Here it is. There we go. Okay, preparation. All right. So liposomes are essentially artificial membranous enclosures. If I were to ask you a definition, that's what I would call them. Okay, they're artificial membranous enclosures, and they're rather easy to make. Okay, so to make them, you have to get a group of molecules that would normally comprise a lipid bilayer. And these would include the glycerophospholipids. These would include the sphingolipids that we've talked about. And generally in making liposomes, those are the 2 things that people put together. They don't mess with cholesterol or anything like that, at least as far as I know. All right. And the idea being that, left alone, they will sit and associate with themselves. But if you force them to interact with water, they will spontaneously form the lipid bilayer that we see. And that's really based on their chemistry. When you force them to interact with water, those hydrophobic regions, the hydrophobic regions of the glycerophospholipids and the sphingolipids don't want to associate with water. So they actually orient themselves, and that lipid bilayer arises as a result of that trying to avoid water. So those hydrophobic things are their geometry is perfect for being able to get on the inside and then leave the hydrophilic phosphates and things like that on the outside. So they arrange in that way. So if we shake it up, or in this case sonicate it, which is another way of doing that, we get them out of their comfort zone, and they go out here and they start making these structures. Well, the structures are random in how they form, so some of those structures will contain, if we have molecules in this solution, some of those structures will contain the thing that we had in the solution to start with. Well, the beauty of that is that it's just as these guys are relatively easy to make, so too are they relatively easy to fuse with natural membranes. And so by sort of forcing them together with natural membranes, they will in fact fuse, and when they fuse, they dump inside of the cells the thing that we had inside of them. Okay?
Is this a way for cancer treatment?
Yes. Is it a way for cancer treatment? It is a way to get cancer drugs inside of cells. If we have time later in the term, I'm going to tell you about a novel way of treating any kind of cell that you want to kill. Okay, I'll briefly tell you here, just since you've asked the question. Okay, so there's anybody ever heard of AVI Biopharma here in Corvallis? Nobody's heard of it? Okay, it's the biggest biotech company in Corvallis. It was actually created by a person who was a postdoc in this department back in the 1970s. And he had a very simple idea that he said was going to cure every disease in the world. And his idea is simple enough that it will do that. Okay? So he spent, I don't know, probably 10 years literally doing research in his garage. All right? I'm not making this up. Making these compounds that would literally target anything that you wanted to kill or stop. Okay? I'll tell you how they work in a second. So after a long period of time, he got some investors, and they saw the wisdom in this, and they got a bunch of patents, and they actually have some things that are currently in testing. All right? They're perfect on paper. The problem is they will only work inside of a cell, and getting them across a cell membrane is a challenge. So they've got some strategies for doing that. How do they work? Well, everybody knows what messenger RNA is, right? So messenger RNA contains the coding for protein. His idea was, well, messenger RNA is only one strand, and if I know the sequence of that strand, I can make a complementary sequence that'll pair with it. It's called antisense. Okay? Well, rather than make it out of DNA or RNA, which would get dissolved in the cell by the cell's enzymes, he made an artificial one that was like DNA and RNA, but that it wouldn't be destroyed by the enzymes. So it forms base pairs, it forms a duplex with the RNA, just like a regular RNA or DNA would, but it can't be destroyed by the cell. In the laboratory, it works absolutely perfectly, because what happens is you put this in the place where the ribosome would go along and make protein, and it can't make protein, because there's a duplex there instead of a single strand. All right? So if he has a bacterium that he wants to kill, all he has to know is a critical messenger RNA sequence. He chemically synthesizes this artificial sequence, and he can kill him. I mean, it's remarkable. If he has a cancer cell that has a single mutation, and that single mutation is in a critical gene, he can stop that gene from being made. Okay? It's perfect. I mean, it's brilliant. It's absolutely perfect. And he would be a Nobel Prize winner today if this were easily deliverable across cells. Okay? So getting things across cells is a very, very important consideration. You can design a perfect drug, but if you can't get it across cells, you don't have a perfect drug. I guess that's duh. So anyway, that's I find his ideas, and he's a genius. He has absolutely brilliant ideas. He sold out when AVI went public, and then started his own company in Philomath now called Gene Tools, where he makes these synthetic drugs and sells them back to AVI to test. It's a pretty good deal, you know. Anyway, so. He knows more about making money than I do. So, Jody?
I had a question related to the TM point
Okay.
membranes and
TM.
the fluid mosaic model and that whole thing.
Yep.
So membranes can adjust their fluidity sort of over time based upon lengthening, shortening, and saturated versus unsaturated.
Mm-hmm.
So why integrate cholesterol at all if all it does is expand the TM point? Is it that they don't have a fast enough response time?
Yeah, that's a good question. I don't know fully the answer to that question. I wondered that myself. But it appears that expanding that TM range does seem to have some important role in the cell. And I'm guessing it gives more flexibility, and there's something in that transition range that is actually advantageous for cell flexibility, but I don't know the answer to your question.
Are there very many anchored proteins or any at all that actually use cholesterol as an anchor?
Are there any proteins that use cholesterol as an anchor? To my knowledge, I don't know of any, no. Things that use anchors tend to use fatty acids as anchors. So you'll see a lot of them will use myristic acid, which has 14 carbons. And it's a nonpolar thing, and that 14-carbon thing just buries itself in the nonpolar region of the lipid bilayer, and the other portion sticks out and holds on to the protein. And that's what's commonly used for anchors. I don't know of cholesterol being used as an anchor, no. Liz, you had a question also?
Could you do the same thing with I don't know if I'm
With what?
I don't know if I'm pronouncing this correctly, but micelles?
Yeah, so micelles, no, you really couldn't, because micelles don't form that enclosure in the same way. If I have micelles on here or not.
Under characterization.
Yeah, okay, there's a micelle. Okay, so you'll notice that with this, there's really nothing to put in here, all right? And it's not the same as the lipid bilayer, so we don't have an aqueous environment in here, essentially. We have these tails are all just associated with each other. Would this be a way to carry in something nonpolar? Perhaps. But I'm trying to think. I'm not sure how well this would fuse with the membrane. That would be the one consideration. Because, for example, this would be like what a detergent does. And when we wash our hands, we don't integrate that detergent into our membranes to any significant extent that I'm aware of. So I don't think that would go. Yes?
Back to the pumps.
Yeah.
So there's the sodium potassium pump.
Yep.
And then calcium is pumped out due to the sodium being pumped in.
So, the sodium, the sodium calcium pump. Yes. Uh-huh.
Yeah. So are those pumps just like completely unrelated? Like the amount of, are they in different cells, or how does potassium sodium pump not affect calcium?
Oh, okay. So that's a good question. So how is it that? Okay, so let's start with the sodium calcium pump. Okay, so the sodium calcium pump uses a sodium gradient to drive out the calcium that's inside of a cell. So it's an antiport, sodium in, calcium out, right? And your question is, if I have a pump over here that's pumping sodium out, and I have one over here that's letting sodium in, are they at cross purposes? And the answer is no. Okay? What the sodium calcium pump is doing is taking advantage of what the other pump is doing, just like what a nerve cell is doing in taking advantage of that sodium concentration gradient. So cells are extremely opportunistic, and this is a prime example of that. Another place where we see this opportunistic thing is if we look at bacterial cells with respect to the lactose permease. You saw that protons on the outside were carrying in that lactose. How do we get protons on the outside? Well, bacteria don't have mitochondria. They kick protons out of their cell. And so those protons are carrying lactose back in. So yeah, cells are really advantageous at that, and that's not they're not going to cross purposes at all. Jody?
Is the sodium-potassium pump and the sodium-calcium antiport, is the sodium-potassium pump able to sort of outrun the other one since it's a 3-to-2, whereas the other one, isn't it a 1-to-1 sodium-calcium?
Okay.
I guess I'm just asking kind of relative rates.
Okay, so he's asking about relative rates. So he points out that the sodium, I'm repeating this for the people who didn't hear what you said on the TV. So the sodium potassium pump pumps out 3 sodiums, and the sodium calcium is letting in 1 sodium for each one. But we haven't said anything about how fast either one of these work. So I would be reluctant to say one outruns the other just because based on how many that they're there. I would agree that if the rate of pumping of one was the rate of movement of the other one, that yes, the sodium potassium would win. Okay? But you have to keep in mind that cells are striking a balance here. They're not doing a race. And that balance is rooted in that osmotic pressure. So once they get it to that point where they want that osmotic pressure, they're going to slow down. They're not going to keep pumping it out. Okay? That would be completely counter to their purposes in doing that. Just like in the mitochondria where we see the proton gradient gets very high, okay, the mitochondrion can't keep pumping protons out even if electrons want to flow because the gradient's too high for it to do that. So that's really the determinant that's there. So it's not really, as they say, a race with any of those. Does that make sense?
So at that point, would they just scale back on the transcription of whatever created those particular pumps and allow membrane levels to fall a little bit?
So would they slow down the transcription of those pumps and let them fall? I suppose ultimately that would happen, yes, but that would be a longer-term proposition. When you affect transcription and translation, usually, especially in eukaryotic cells, those are longer processes. Okay? Yeah?
She said it was the osmotic pressure that
Yeah.
controlled the speed with the diffusion pump.
Mm-hmm.
Do they have a way of sensing that pressure? Or?
Yeah, do they have a way of sensing that pressure? And that's a good question also. The way they have of sensing that pressure is actually with the electrochemical potential that's there. And so that does, as we've seen, that was why I was sort of drawing the analogy to the mitochondria. We see that potential gets very high, and they don't keep pumping protons out because they just simply can't do it. There's a similar thing that's happening with that sodium-potassium potential as well. That gradient gets high enough, it's not going to fight that. Yeah. Yeah, Liz?
Is the word pump synonymous with active transport?
Yeah, is the word pump synonymous with active transport? And the way I use it, yes, it is. Absolutely. Yeah. Whenever I say pump, I'm thinking of an active transport process. And when I'm thinking of an active transport process, what am I thinking of? At least one thing is always moving against the gradient. Yeah, that's what active transport is. Yeah.
Could you talk about the mechanism of the iron copper peroxide bridge?
Yeah. Did I talk about the mechanism of the iron copper peroxide bridge?
And like what it's for?
Yeah.
Exactly.
So let me show you. That's complex 4. So let me pull that up and show it to you. I haven't said anything about it relative to mechanism. No. Okay. And part of that reason for that is, as I understand, I'm not an expert in this field, but as I understand it, this peroxide bridge itself is a bit controversial. It used to be accepted as pretty much fact, and now I've heard that there are other models that are explaining that. So I haven't really talked about the mechanism there. Let's see. Complex 4 is mechanism of action right here. Okay. So here's what you're talking about. And I don't really have much to say about it other than the fact that this is a way of grabbing and holding on to oxygen and providing a place for the electrons, excuse me, a place for the electrons to come and act. Okay? So because it's held in this configuration in the confines of this complex, the 2 protons and 2 electrons can come here, and then further 2 more protons can come over here and release that. Okay, so we've had 2 electrons here, we had 2 electrons here for a total of 4 electrons. But I haven't talked about how this goes to this, goes to this. And this actually is a good and important question because it's actually in the mechanism by which this occurs that we see the reactive oxygen species being produced as a byproduct. All I've said in class is that they can be produced as a byproduct, but I haven't said how. And that's actually beyond the scope of this class, to be honest with you. Yes?
So electrons come in one at a time through complex 4.
Electrons come in one at a time through they actually come from cytochrome C, but they're being dropped at the complex 4. That's correct.
So for all the other complexes, do they come through in pairs? Or?
Okay, so electrons come into the electron transport system in pairs. So NADH and FADH2 drop off electrons in pairs. So complex 1 and complex 2 both accept pairs of electrons. They dump off pairs of electrons to coenzyme Q. But coenzyme Q passes them off one at a time in the Q cycle through complex 3. So everything after coenzyme Q is 1 electron at a time. Okay?
Could we take another look at the 2 stages of the Q cycle?
Sure. Q cycle, yeah. So Q cycle, I think is pretty cool, actually. I can't make that go over. Okay. Can I shrink it? No, I can't do that. Okay. Well, for some reason, that's running off the side over there, and I don't know why. Well, actually, hold on. I'll bring it on. Does that bring it on? Oh, maybe not. Okay, let's try this then. That didn't. Oh, there we go. Okay. Now, I think we've got it on there now. All right. Okay. All right. So there is our Q cycle. All right. So let's orient ourselves. This big guy here is complex 3. So all the action of the Q cycle is happening in complex 3. And this Q pool simply means the mixture of Qs with 2 electrons and Qs with no electrons. Both are necessary for this cycle. All right? So we start this process with an empty complex 3. All right? So this is after we've loaded it. We're starting with an empty complex 3. In the very first step, we can imagine that an empty Q, that is with no electrons, binds down here. Okay? And then we have a QH2 at back up here. We have this empty. We start with a Q down here, and we have a cytochrome C up here. Okay, we get a QH2, that is a Q that has 2 electrons that comes in. At this point, we're ready to start this process. So to get it started, we have to have a cytochrome C, we have to have a Q with 2, and we have to have a Q with none. So that split happens, as I've talked about here, 1 electron going to cytochrome C, 1 electron going to this other Q to make a Q with 1 electron. Okay? And that leaves this Q that started out with 2 with no electrons and no protons. The protons are kicked out. This guy goes back to the Q pool. In the next step, notice this guy stays in here. It hasn't gone anywhere. It stayed in the confines of complex 3. And a Q with 2 electrons comes in. The Q with 2 electrons and a new cytochrome C. Don't forget, we have to have a new cytochrome C because this guy can only handle 1 at a time. So we brought another one in that had no electrons. And this guy does the same thing that this guy did. 1 electron here, 1 electron here. And when that happens, 2 electrons have been transferred. 2 more protons got pumped. This Q goes out, and this Q goes back out. Okay? So now we're back with a QH2. We've moved 2 electrons, which essentially means we have converted 1 QH2 to 1 Q. Connie?
It almost feels like the Q, the center, coenzyme Q doesn't even give its electrons to complex 3. It just kind of uses complex 3 to give it.
Exactly. It doesn't. You're quite accurate in what you just said. It doesn't give its electrons to complex 3. Complex 3 is basically a dock for all. It's like a network center for these things to come together and do their thing. That's exactly what it is. Okay? Yes?
So protons are pumped out of complex 1, 3, and 4?
Protons are pumped out of 1, 3, and 4. That's correct.
How come they're not pumped out of complex 2?
I'm sorry?
How come they're not pumped out of complex 2?
Well, there's not enough energy there to do that, and that certainly hasn't evolved that way. So if there were the energy to do it, then we would get that. It turns out that getting electrons off of FADH2 actually takes some energy, and so that energy isn't available as a consequence for the pumping of protons.
And that's why FADH2 doesn't produce as many ATPs.
And that's why FADH2 does not produce as many ATPs, because not as many protons get pumped. Exactly right. Yep. Yes?
So is cytochrome C pretty much just like an electron acceptor?
So, well, cytochrome C is an electron transporter because it's accepting electrons here, but it's donating them to complex 4. So I like to think of it as a shuttle. It picks it up at 1, it drops it at the other. It picks it up at 1, it drops it at the other. So it's back and forth, kind of like the ping pong mechanism of the enzyme, right? On the one side, it's got electrons, and the other side, no electrons. Electrons, no electrons. Yes, sir?
So this is all happening in lipid bilayer?
This is all happening in the intermitochondrial membrane, which is a lipid bilayer, yes, but this is happening in the intermitochondrial membrane.
So where does cytochrome C is that
Yeah.
matrix, or is
That's-
that in the membrane as well?
Okay, so cytochrome C, it's a very good question, is a peripheral membrane protein. So it doesn't extend all the way through, but it's linked with that one layer of the lipid bilayer, and it's because of that it's very mobile. If you think about this guy is acting as a shuttle. It has to move quite a bit between complex 3 and complex 4, and so the more mobile it is, the faster those electrons can get moved. Okay? That's also a consideration for coenzyme Q. And coenzyme Q turns out to be a very small molecule. So small molecules can move very rapidly in the lipid bilayer, okay? And that works out well. So coenzyme Q is bringing things between complexes 1 and 3 or 2 and 3, and cytochrome C is taking things between 3 and 4. The big complexes don't move very fast. And so it's really nice to have those little shuttle things moving things quickly between them. Yes, sir?
I may be getting my terminology messed up here, but is coenzyme Q also referred to as ubiquitin?
Coenzyme Q is also referred to as ubiquitin. Yeah. Actually, back up. It's not called ubiquitin. It's called ubiquinone. There's a difference. Yeah, ubiquinone. I make the mistake myself sometimes. Yes?
You said that FADH2 doesn't have as much energy as an ADH, but back in, is it like E2 or E3 when you're trying to recharge, what was it?
Yeah, I know exactly what you're going to say. That's a very, very good observation. Okay, so what Connie, let me show you what you're getting ready to ask, okay? It's rare I have somebody notice this, but it's a very, very good observation. So let me get my, come on here. Nope, that wasn't what I wanted. Green's what I want. Okay. So what Connie's asking is, she's noted that when I talked about this reaction scheme here, look what happens. This guy is donating electrons from FADH2 to NAD. And I just told you something contrary to that. I said that it was harder to get these electrons and protons off than it was off of this guy. So this guy shouldn't be able to donate electrons there. Okay? Anybody know why or how it does it?
Is it because it's part of that enzyme or that complex enzyme?
Exactly. This guy is covalently linked to the protein. And the electronic environment that this is in is different than that of a free FADH2. So we call it FADH2 because it has that ring, but it's actually covalently linked to the protein.
Okay.
And that's not the same as a FADH2 that's floating around.
Okay.
Yeah. Very rare I have somebody notice that. That's a very good question. Yes, sir.
Speaking of FADH2, well, I guess speaking of the shuttles in the electron transport chain, is there movement between the complexes more or less random and from fusion, or because it seems like they're not specifically linked to a protein that is moving them directly to another complex, or maybe I'm mistaken.
I'm not sure. I missed the first part of your question.
Cytochrome C, for instance, moving the electrons from complex 3 to complex 4.
Yeah.
Is that random?
Oh, is the movement of the complex random? Yeah, it is. Yeah, it's random. Now, keep in mind that there's a lot of protein in this membrane. Remember I said the inner mitochondrial membrane is loaded with protein. So that random movement has to have advantages of proximity if it's going to be effective. And that's one of the reasons it has a lot of protein in there. So it's much more likely it's going to bump into the right thing and donate that electron. But that's exactly it. I need to make a phone call, folks. I hate to do this, but my pie is going to disappear if I don't. And I just can't let that happen. So I would never do this in class, but let me just do this real quickly. They'll like the fact that I'm doing this in front of a review session. I can have you guys say hi to them. How about that?
Give them a link. They can watch it online.
I'm curious what sort of pie is it?
Me too.
Tell them where you're at. We'll wait.
Bring some up here for the whole class. Jessica, how you doing, kid? Well, I'm not avoiding you, but you're not going to believe where I'm at right now. No, I'm standing up in front of my BB451 class giving a review session, and I couldn't respond to your thing. And I said, you know, there's a pie at stake here. So the class said, hell yeah, get on there. So here, so twist my arm. I'm in ALS. Oh, well, okay, come on up. We're in 4001. Maybe people will help me eat it. Or if the exam doesn't go well, they may throw it at me. Oh, okay, so I'll see you in a bit. Take care. Bye. I've never done that before. That's weird, but it's for pie, folks. I mean, come on, Giovanna.
Okay, yeah, I think this is probably a stupid question, but how does it do with the mitochondrial membrane?
Yes.
So we have the DHAP, and we have the cytoplasmic 3 G3P dehydrogenase.
Yes.
Why is it going? Because usually when you have a dehydrogenase, you always have production of NADH. Here instead, you're having the production of NAD.
Okay, so you're talking about the shuttle system, right? Okay, so let me go to that shuttle
And then
system.
the same terminology in the other hand, it goes in the normal direction.
Right. Okay. So let's see. So the shuttle, right here, is this the one you're talking about?
No, it's the glycerol.
The glycerol. Okay. So so here's your shuttle, and your question was, why are we making NAD?
Right.
Okay. Well, remember that what we're trying to do here is we're trying to move electrons from NADH into here.
No, I mean, I understand why that's happening. It's the name, because every time we have
Oh.
it's very confusing. In that situation, you're making an NAD while in the opposite side.
I see your question. Very good question. I hadn't even thought about this. Okay, it's a very good question. It's named for the reverse reaction. right? So if we go that way, we make an NADH. I hadn't even thought about that before, but that's the reason why. Yeah. But it is a reversible reaction. That's what's happening with it. Yep, sure. Yeah.
So you get less energy from the electrons because you're transferring it to FAD. Why wouldn't it just transfer into NAD?
There's no NAD to transfer it into. So the NAD is in here.
Oh,
So in other words, this guy can't cross the membrane, and neither can the electrons.
so NAD can't be attached to an enzyme either?
NAD is not attached to anything out in the cytoplasm. This is you got two choices. Right. But I'm saying that there's nothing that attaches to NAD like that. NAD is free in the cytoplasm. There's no attachment. There's no shuttle to get it in. I mean, there's no mechanism to get it in, but there's only this shuttle that's there. Okay? Yeah.
So is FAD giving its electrons to coenzyme Q, or is that Q just?
Yes. So what's happening here is this is accepting the electrons here, and then you're seeing the other reaction where this is dumping its electrons to coenzyme Q. Exactly. So what's happened is this guy has gone from electrons at NADH to electrons at coenzyme Q, but we bypassed complex 1. So we didn't have any pumping happening from complex 1. Okay? Liz?
In the citric acid cycle, when you kick out a GTP,
Yes?
does the cell use that GTP like equivalently to an ATP that might be
Yeah.
made from?
So the question is, when the cell makes GTP in the citric acid cycle, does it use it in the same way it uses ATP? And the answer is yes. Exactly the same energy. And GTP is an energy source for protein synthesis. So it's a very important energy source. Absolutely. Yeah. And there's other ways of making GTP. So this isn't the only way cells have of making GTP, but it's one. Yes?
Is there a use difference for GTP and ATP, or do they kind of interchange?
Is there a use difference, meaning some reactions use it, some others don't? Yeah, there's definitely a use difference, yeah. So in protein synthesis, we don't see anything except GTP that's used. You can't substitute ATP. And if we look at, let's say, hexokinase. So hexokinase, you remember, used ATP to put a phosphate onto glucose. GTP won't work. So in essence, those aren't barriers that get crossed. Enzymes have evolved one or the other. Yeah. Yeah.
Just to clarify, did you say that this one was involved in insect muscle?
This is involved in insect muscle. Uh-huh.
Okay, and then the malate aspartate one is in us.
In us. Uh-huh. Good questions tonight. I told Indira, I said, oh, they'll probably have a couple of questions. There's no problems to solve. So there's been really good questions tonight. Connie, you have a question?
Just totally random, but for the sodium glucose pore, do they, since they come in together,
Uh-huh.
do they just go through like one channel, or are there two separate channels going for sodium?
Yeah, somebody asked me that after class the other day. With the sodium glucose channel, are there two different channels that are used? I don't know the answer to the question. It wouldn't surprise me if you didn't have a separate one for each one, but I don't know the answer to that. They are very different. One's charged, one's not. One's a ring, the other is not. So I would think that there would be separate ways of doing it, but there also may be an association that's made between them that brings that through. So I just don't know the answer to that. Yeah. Yes, sir.
Referring to the conversion between pyruvate and acetyl-CoA.
Uh-huh.
I believe you mentioned that the decarboxylation is important. The independence of the decarboxylation is important from the oxidation.
Uh-huh.
For fermentation.
Yes.
Or implications. Could you go over that?
Yeah, sure. So the question had to do with the ability of the decarboxylation of pyruvate to be independent of the oxidation, and is that important? And the answer is yes, it is. So if we look at that mechanism shown right here, all right, we see, as I noted, a decarboxylation first, followed by an oxidation, followed by transfer to get acetyl-CoA. And this is where I called E1, E2, E3, and people didn't like that. Okay, so that was my mistake. Now, your question has to do with, is it important that, hey, there she is, there's my famous pie. No, this is cool. I just want to make sure they don't throw it at me. Thank you, Jessica.
You're welcome.
This is one of my former students, and she is currently at OHSU in her third year of medical school.
Yeah.
Yes.
Hooray.
And she didn't throw it at me.
This matters.
Good. Thank you, Jessica. I'm sorry I can't get together with you guys tonight. So if you try to call Indira, she's giving an exam right now. So both of us are, a little tied up. Sorry. Thanks again. Take care. Okay. So the significance of the independence here is that because this can happen independently, all right, there's no NAD that's needed. Now, the significance of that is since no NAD was needed, we can use the product of this guy and then take electrons off of it and I'm sorry, take the, use the, let me back up on that. We can take the product of that, which is basically acetaldehyde, and we can reduce it with electrons from NADH to make NAD. If we were using NAD, then it'd be an even trade. Right? So the significance is that this now provides a substrate that will accept electrons and allow yeast to make NAD. And of course, when they're doing that, they're making alcohol. That's when oxygen is not present. That's right. So like in any cell, when oxygen is not present, NAD is limiting. And so you have to have other ways of making NAD, and this provides a way for them to do that. Yeah. So can I repeat that? So to repeat that, what I said was the significance of the decarboxylation being independent of the oxidation is that this making of this molecule does not require NAD. If they weren't that way, then this would require NAD and it would produce NADH. Then if the cell reduced that, you would use an NADH. Okay? You would use an NAD and you would make an NAD. There'd be no net gain. But if it sidetracks here, it sidetracks before the oxidation happens, and it's able to reduce it. And so now you can have a net synthesis of NAD if you go down here and do the fork in the road. Does that make sense? No? Is that a yes or no? I see some people saying no. So let me, okay. If I go through here, if I go all the way over here, all right? If I don't subvert that, this requires an NAD, okay? To go right here at this step, something's got to take those 2 electrons, right? Okay? Something's got to take those 2 electrons. NAD would take those 2 electrons and make NADH. So if we go this route, which is what we go through, there's no way of making NAD. You can't do it, all right? But if you stop it here, before the oxidation has happened, which is what his question was, is there significant that this is separate from this? Yes, it is. Because if we stop it here, we can run down and add electrons from NAD to this to make ethanol. That's what yeast are doing. And when they're making ethanol, the reason they're making ethanol is that generates NAD, which keeps their glycolysis going. Okay? Yes?
I thought that the electrons from the oxidation went to FAD, and then FADH2, and then to NAD. Is that?
No. So I'm not sure what you're saying. You're talking about you're talking about here in the mechanism? Yeah. Well, ultimately they do go to NA. Ultimately, you make an NADH. Right. Yeah. Right. So that's 2 more steps after this, but the product is NADH, not NAD. Right. Yes, sir.
Can we look at how the ABC transporters are different from the P-type transporters a little bit?
Well, I haven't said anything about how they're different, other than the fact that his question has to do with the difference between the the ABC transporters and the P-type transporters. But to address your question, what I did say about the P-type was that they involve the phosphoaspartate, and that's the only thing I said about them. The ABC transporters simply don't involve a phosphoaspartate. Okay, so that phosphoaspartate is a covalent intermediate that's transient. It's like the serine protease, where we have the reactive alkoxide that does the attack and does its thing. In the case of the P-type ATP transporters, they have, to show you the mechanism, they get that phosphoaspartate side chain in the middle of their mechanism that helps to drive the overall process. And that is shown here, I think. Yeah. Okay. So here is the chamber the transfer of the phosphate from ATP to the transporter puts a phosphoaspartate there. That causes a charge change in the protein. It's a driving force for the shape changes that happen. And the shape changes that happen result in the movement of the calcium out, the release of the ADP. Then in the next round of the cycle, the removal of that phosphate causes the system ultimately to get back to its original state. So we have two states that arise from that phosphoaspartate.
So in step 5, it releases that orthophosphate, but where does that water that comes in end up at?
Where does it end up at? So when you release a phosphate, one of the protons goes here, and the OH goes here, right? That's what a hydrolysis reaction will always do. Okay? What's that?
It's a Monday.
It's Monday, yeah. Yeah.
Could you explain how the ABC transporters work?
I haven't explained how they work. I just simply said that they were another transport system. I used to go through all the transporters, and I would show the steps in all of them. And to be honest with you, A, I'm not a mechanism person, okay? And B, it's really hard to ask mechanistic questions like that without just simply regurgitating the whole thing. And I just don't see that that's really very useful. You saw as much mechanism when I talked about serine proteases as I will talk about. And I talk about it there because I think it's important to know a catalytic mechanism and how it occurs. But mechanisms in general like this, I mean, what can I say? There's a shape change, you know? I mean, that's really what's happening in all of these things. And it's really hard for us to conceptualize that and sort of do something with it on an exam. So I don't really talk much about them anymore for that reason. Yes, Connie.
How is succinate dehydrogenase different from other dehydrogenases? I think you mentioned it.
How is succinate dehydrogenase different from other dehydrogenases? Well, it's different in the sense, only in the type of reaction that it's catalyzing. Okay? So succinate dehydrogenase is an enzyme that is taking electrons directly off of a carbon-hydrogen system. Okay? The other dehydrogenases that you've seen in class up until this point have involved oxidation of an OH to make a ketone or an aldehyde. You've also seen decarboxylations that have happened of alpha keto acids like alpha ketoglutarate and so forth. But you haven't seen something take protons and electrons from a single bond like that. All right? And that takes something with greater pulling power to get those electrons out, and that's why FAD is used. So it's the FAD, I would say, that's more interesting and more unusual than is the enzyme itself, because the FAD provides that pulling power to get those guys off of there.
Repeat that one more time, sorry.
I'm not sure what to repeat.
Just the type of dehydrogenases we've seen in the past.
Oh, well, the type of dehydrogenases you've seen in the past have been things that converted an alcohol to an aldehyde or a ketone, or they did a decarboxylation. But you have not seen dehydrogenases that are acting on a bond system like this. Now, this one is not unique. Okay? We'll see when we talk about fatty acid oxidation that the very first step in fatty acid oxidation involves an oxidation just like this, and it uses FAD for exactly the same reason. These are hard to get protons and electrons off, and it takes the pulling power of that FAD to make that happen. The second step of fatty acid oxidation is exactly like the malate. Is exactly like going from fumarate to malate. You add water across a double bond. The third step is just like malate going to oxaloacetate. You're converting an alcohol to a ketone. So these reactions that you see here in the citric acid cycle, you will see again when we talk about fatty acid oxidation. Yeah?
Just going off that last question, I think what you said about the succinate dehydrogenase was that it was found in the membrane in mitochondria.
It is found in the membrane, and others are generally soluble. That is a difference of the enzyme. That's correct. But there are other enzymes. You've seen, for example, adenylate cyclase. Last term, that was embedded in the membrane. So there are enzymes that are, in fact, embedded in membranes. That's not that unusual. Good questions. Connie?
Can you go over pyruvate dehydrogenase? I know there was like a kinase and a
Okay.
phosphatase.
Okay. Pyruvate dehydrogenase regulation is, let me think about this for a second. So I talked about that relevant to,
Under cycle regulation.
there it is, yes, I knew it was there somewhere. Okay, so with this enzyme we have a couple of reaction mechanisms. One is that when we phosphorylate the enzyme is inactive. When we dephosphorylate the enzyme, it is made active. That's just simply a kinase phosphatase type reaction. That's not, to my knowledge, protein kinase A. It's a different kinase that's doing that, I believe. I'm pretty sure that's right. And the other consideration is that the enzyme, because it is using NAD, it is sensitive to NAD concentrations. Okay? And these factors negatively affect it: ATP, NADH, and acetyl-CoA. And acetyl-CoA being a product, not surprisingly, is going to have a negative effect on the enzyme. So, but again, what I said about regulation, I really want to emphasize, and we think about the regulation of not only this enzyme, but also the citric acid cycle, the biggest consideration is the amount of NAD. That's what it comes down to, NAD and FAD. All right? Yeah, phosphorylation is a factor. We haven't talked much about it this term, and we won't. All right? But the biggest factors for us to consider are the oxidation state of the cell. The cell has plenty of NADH. This ain't going to go. Yes, sir.
We talked a little bit in class, and then we read in the book about the different levels that NADH and FADH2 have relatively for pumping protons across the membrane to contribute to that gradient. Which did you want us to use?
I'm not sure I understand the...
Because NADH and FADH2 contribute the electrons that result in protons being pumped across the inner mitochondrial membrane.
Right.
But they contribute in disparate amounts. One of them contributes more.
Right.
I believe the book said it was 2.5 to 1.5.
I said 3 and 2.
3 and 2.
That's what I've used. And again, I'm not. This is not about getting hung up on numbers there, but you can see that there's more protons pumped if it's coming from NADH than it's coming through FADH2. Right.
And the accepted range overall for ATP generated is between 30 and 38.
That's right. So 30 and 38 encompasses pretty much what people will all agree on. Yes. Yes, Connie.
You made an anecdote that like carbons are only lost after the second round of the citric acid cycle, or something like that.
Okay.
I don't know if I missed.
Well, carbons are lost in every round. I think what you're thinking about is where I, as an aside, said the carbons from acetyl-CoA that come in aren't lost in the first round. Okay, so let me show you the cycle. So where's the cycle here? Here. Nope, that's not it. Which one? This one? Yeah, okay. All right. This is color-coded. Here's an acetyl-CoA coming in. They're in green, right? All right. There's the green. There's the green. There's the decarboxylation. You haven't lost any green, right? There's the green after the second decarboxylation. So in the first round, you didn't lose any of the carbons of acetyl-CoA. Only on the next time around will you lose them.
Okay, so it just chews up the opposite ends each time.
Well, they're just not in a position to get decarboxylated the first time. It's not a major point. As I said in class, and that's the reason I made it an aside, was some people get really their panties in a wad about this. But I mean, to be honest with you, it's okay. That's nice, but so what?
Can we talk about neurotransmitter recycling?
Sure. Neurotransmitter recycling, I think, is actually kind of interesting, although I just mentioned it with respect to cocaine addiction. But I think it's interesting to think about. So let's see, membrane transport here. And when we see here, okay. So this is how nerve cells talk to each other. And that communication between nerve cells is obviously important for that information to get transmitted. That happens when this voltage wave that's moving down here that happens from the movement of these sodium and potassium ions comes in. That voltage wave causes these synaptic vesicles, which contain the neurotransmitters, to fuse with the membrane and release the neurotransmitters. So the neurotransmitters are this guy right here. The binding of the neurotransmitters, the exit of the neurotransmitters here cause them to bind to this nerve cell and stimulate another wave of sodium and potassium moving through here. And normally these guys would have to be recycled, and normally they are recycled just fine. But in cocaine is one example of a drug that in some case, with some neurotransmitters, interferes with their ability to be reabsorbed. And it tends to be associated with pleasure centers. And so when that happens, then the cocaine enhances that pleasurable feeling by leaving those neurotransmitters stimulating this pleasure neuron right here longer. Okay?
Is this supposed to be, is this I guess, a one-way street? Like your neurotransmitter going from the red into the green, it can't go from the green back into the red?
It's actually not going into the green. It's binding to the green. Okay?
Oh, and then, okay, so it's not.
It's not entering the green. No. Okay. So you see it's being released here. And so it's exiting here, and it's going to bind to receptors on here. And then when this thing's done with them, it lets them go. Well, if it lets them go and they're not getting recycled, they can come back and bind and bind and bind. So it prolongs that good feeling. That's why it gives people that high. Yeah. Make sense?
Yeah. Isn't that one of the targets of neurotropic medication like SSRIs do?
Don't know. I don't know.
Reuptake inhibitors that prolong.
There are reuptake inhibitors that are used in treatment, but I don't know. Again, it depends on the neurotransmitter and the neuron itself that's being affected, Jody. So I'm not a medical doctor, so I can't tell you that. I don't know. All right.
I actually have a yes/no question.
A yes/no question. The answer is no.
Well, the question is, does arsenite prevent lipoamide from reforming?
Does arsenite prevent lipoamide from reforming?
Like,
What does that mean?
well, once lipoamide becomes like its sulfhydryls,
Yes.
it needs to like reform before it can get used.
Oh, good question. Okay, so I'll ask everybody else that question. So the product of the treatment of arsenate, and where was that? That was, my phone was going nuts here. Let me think, where was that arsenite? That must have been here, right?
Was it arsenated valve?
Right. Second from the bottom. Yes, okay. So your question is that we have this guy right here, but for it to be active, it's got to be in the disulfide state, right?
Right.
Well, what happens to these things in cells? We learned that last term. They're readily oxidized. That is, it doesn't take much to oxidize them. You put them in close proximity, they will readily oxidize, and if they do that, you've made a disulfide again. So the answer is nothing extra would be needed to do that, no.
Oh, okay.
Okay?
Okay, I guess I just got confused.
Clear as mud? All right, guys, that's a long one, so I will get the video posted as quickly as I can. And thank you for a bunch of good questions, and thank you for letting me talk to the pie lady. This is my treat.
If you're a medical school, make sure it's not a meat pie.